The information coded in the DNA of our genes is transcribed into messenger RNA, which is then decoded and translated by ribosomes into proteins. In order to understand the nature of the genetic code and how it works, we must first learn about the polymeric structure of proteins. Proteins, like DNA and RNA, are linear polymers. Monomers that make up proteins are called amino acids, and they come in 20 different flavors. Each amino acid has a central carbon atom called carbon alpha. An amine nitrogen group is linked to one side of carbon alpha, and a carboxyl group, a COOH acidic group, is bound through another valence of carbon alpha, hence amino acids. The remaining two valencies of carbon alpha are bound to a hydrogen atom and to any one of 20 different side chains or residues designated R groups. The 20 different R groups can be divided into 4 types that correspond to their chemical nature: hydrophilic, hydrophobic, negatively charged, and positively charged. The following table shows the 20 amino acid structures divided into these 4 categories. You don't need to learn the precise structure and composition of each amino acid, but I suggest that you examine them and see how the R groups correspond to their chemical classifications. Proteins typically contain hundreds of amino acids, and their composition, as well as the order in which they are built, dictate how a particular protein will fold and function. Obviously, if a protein is to function in our blood, it must be soluble and thus hydrophilic. It stands to reason that many of its amino acids will be hydrophilic and even positively and negatively charged. On the other hand, many proteins function as part of the cell membrane and therefore must contain hydrophobic regions that can associate, bind, and even traverse the membrane lipid bilayer. Proteins are the major components of our cells and the organs that make up our bodies. All our enzymes are proteins. Many of the structures like hair and cartilage are made of proteins. Our muscles are made of special proteins that enable movement and contraction. Ultimately, our well-being is dependent on the activity of proteins. Mistakes in their production, called mutations, can sometimes lead to disease. Diseases can also develop when proteins get damaged. The amino acid monomers link to one another through a condensation reaction, just as all the polymerization reactions we've studied so far. Here you can see that the flanking either side of carbon alpha, there is an amine group on the left and an acid carboxyl group on the right. Removal of a water molecule derived from the hydrogen of the amine group and a hydroxyl of the carboxyl group forms a bond between the two monomeric amino acids. This bond is referred to as a peptide bond. Note that the dimer consisting of 2 linked amino acids retains an amine on one side and a carboxyl on the other, much in the same way as DNA and RNA maintain a 5' end and a 3' end. Indeed, proteins are directional, with an N terminus at their beginning and a C terminus at their end. Polymerization of proteins proceeds by adding incoming monomers always to the C terminus. Synthesis of proteins is done by the ribosome, which systematically reads the instructions coded for in messenger RNA, and then, one by one, links incoming amino acids to the C terminus of the protein being made. In the coming unit, we'll discover the exact nature of the genetic code stored in our genes and read by ribosomes.